in single nucleotide substitutions, like change in single nucleotide, silent mutation, missense mutation, nonsense mutation, and frameshift mutation. Single nucleotide substitutions or change in single nucleotide are repaired by DNA polymerase and DNA ligase. There are two types of nucleotide mutations, transition and transversion. One is transition and another is transversion. In transition, the purine is changed into purine and pyrimidine to pyrimidine, purine to purine and pyrimidine to pyrimidine. And in transversion, purine to pyrimidine and pyrimidine to purine can be changed. In silent mutation, codes for same amino acid often involve third position of codon tRNA verbal. Codes for same amino acid. In silent mutation, it codes for same amino acid. In missense mutation, it results in changed amino acid that has chemical structure. Example, it is in missense mutation is in sickle cell disease. Nonsense mutation results in stop codon. There are three types of stop codon: UAA, UAG, UGA. So we remember this: UAA, you are away; UAG, you are gone; and UGA, you go away. Frameshift mutation happened in Duchenne muscular dystrophy. Duchenne muscular dystrophy. Tay-Sachs disease and cystic fibrosis. And in cystic fibrosis. In Frameshift mutation, the nucleotides are deleted that are not divisible by 3. Misreading of all nucleotide downstream protein may be shorter or longer. In splice site mutation, splice site mutation returned in intron DNA. Splice site mutation it happen in some types of cancers, dementia, epilepsy, beta thalassemia, Gaucher disease, and in Marfan syndrome. And mispairing. Strand mispairing. It occurs when DNA polymerase slips and either inserts or removes one or more additional nucleotides by mistake in an area of repetitive nucleotide. Example, CAG repeat expansion in Huntington disease. Mutations in DNA. Degree of change occurs in silent, missense, and nonsense and Frameshift. Single nucleotide substitution are repaired by DNA polymerase and DNA ligase. Single nucleotide substitution change in single nucleotides they are repaired by DNA polymerase and DNA ligase. Types of single nucleotide point mutations. There are two types of single nucleotide mutations. One is transition and another is transversion. Transition and transversion. Transition in transition only purine to purine, for example, adenine to guanine or pyrimidine to pyrimidine, cytosine to thymine happen. And in transversion, purine to pyrimidine can happen, adenine to thymine or pyrimidine to purine, cytosine to guanine can happen. Single nucleotide substitution, silent mutation, codes for same synonymous amino acid, often involve third position of codon that is called tRNA wobble. In missense mutation, it results in change amino acid called conservative if new amino acid has similar chemical structure. Examples of missense mutation are sickle cell disease, substitution of glutamic acid with valine, change of glutamic acid with valine. It happens in sickle cell disease. So it's example of missense mutation. In nonsense mutation, it results in stop codons. There are three types of stop codons. One is UGA, another is UAA, and third is UAG. It usually generates non-functional protein. Stop the nonsense. These stop codon UGA, UAA, and UAG. UGA, you go away. UAA, you are away. And UAG, you are gone. Now I discuss about the frameshift mutation that is most dangerous of all. It's very important. In deletion or insertion of any number of nucleotides not divisible by 3. Deletion and insertion of nucleotides that are not divisible by 3, or if divisible by 3 splits across adjacent codons. Misreading of all nucleotides downstream. Protein may be shorter or longer and its function may be disrupted or altered. May occur due to slippage of DNA polymerase during replication at repetitive nucleotide regions. Frameshift mutation. Example of frameshift mutations are Duchenne muscular dystrophy, sex disease, and cystic fibrosis. Duchenne muscular dystrophy, sex disease, and cystic fibrosis. And splice site mutation, retained intron in mRNA, protein with impaired or altered function. Protein with impaired or altered function. Examples: rare causes of cancers, dementia, epilepsy, sometimes of types of beta thalassemia, Gaucher disease, and Marfan syndrome. And there are examples of splice site mutations. Slipped strand. Slipped strand mispairing. It occurs when DNA polymerase slips and either inserts or removes one or more additional nucleotides by mistake in an area of repetitive nucleotides. Anticipation occurs secondary to insertion of increased repeats across generations. Example: CAG repeat expansion in Huntington disease. So slip strand mutation, it happens in Huntington disease. Due to CAG repeat.